Hey, good afternoon. Welcome back to my channel today. Today I'm going to talk to you a little bit about a very important concept in the field of X-ray crystallography and molecular structure, and that's going to be that of B factor. What is a B factor? You've probably heard of this term before. If you're watching this channel, I'm sure you've seen it and heard it being mentioned about structures. So what in the heck does it mean, right? So the B factor is also called the temperature factor or the Debbie-Waller factor. And so this is a key concept in interpreting X-ray crystallography data. And it helps you quickly assess the quality and the precision of a protein structure model or a nucleic acid structure model, as the case may be. It can apply either way. So what does it represent? The B factor describes how much an atom appears to wiggle or vibrate around its average position in the crystal structure. So atoms in the crystal are not perfectly still. They do move a little bit due to thermal motion, that's heat, and sometimes due to static disorder, that's multiple possible positions in the crystal lattice. The B factor will quantify, this quantifies the extent of this motion or the positional uncertainty. Mathematically, it's calculated as B equals 8 times pi squared times the mean displacement of U squared. So now what is U squared? U squared represents the mean displacement of the atom. So that means more motion gives a higher B value, right? So now, how does the B factor affect X-ray diffraction? In X-ray crystallography, atoms scatter X-rays. If the atoms move a lot, then that scattering becomes a little bit less sharp. It's a little bit lower resolution, and that reduces the intensity of high-resolution diffraction spots. So high B factors, it's really much higher, you'll get a blurring effect in the electron density map of these atoms. And low B factors, down low, that produces crisper, more defined electron density. So it's kind of like tuning the tune on a microscope, right? You can see a clear picture, you can see a blurry picture, right? And so the B factor acts as a correction for the attenuation of scattering intensity due to atomic motion or disorder. Now it's expressed in ranges of the units are angstroms squared, and the typical ranges. So a low B factor might be about 5 to 20 angstroms squared. This shows a well-ordered, rigid region. This may be found in tightly packed secondary structures or in the protein core. A moderate B factor could be between about 20 and 40 angstroms squared. This is typical for highly flexible side chains or even many solvent-exposed loops in the protein. And high B factors are typically anything higher than about 50 or 60 angstroms squared. This indicates regions of high flexibility, intrinsic disorder, or poorly defined electron density. This could even correlate with missing residues in the protein. They've left the residues out just because they can't even, they're moving around so much, and they just can't determine what the structure of the protein is. What does this have to do with biology, right? So in a biological sense, the B factor provides a lot of insight for protein dynamics. It's really important, even within a crystal. So we can see flexible loops, hinge regions, we can study those areas, or even active sites. And then we can correlate this with molecular dynamics simulation data to see that those regions do indeed fluctuate given more motion. We can study that motion in greater detail. And so we can also compare B factors across a structure, and this helps identify mobile domains or binding-induced conformational changes, such as near an active site, right? So ligand binding will also affect the B factor as well. In molecular modeling and docking, regions with high B factors typically correspond to structural uncertainty or induced fit flexibility involved in ligand binding. So this also helps us study proteins in drug discovery to look at those effects. So the key takeaway here, the B factor doesn't just tell you about crystal quality, it really tells you all about atomic motion and structural dynamics within the protein. It bridges the gap between the static crystal structure and the dynamic reality of biomolecules in solution. And so that's B factors, right? Now, so what do B factors? Let's look at this. We do have NMR structures in the protein data bank as well. Do NMR structures have a B factor? No, they do not. B factors are X-ray specific because there's two different things, right? The B factor is specifically a temperature factor in X-ray crystallography that quantifies how much each atom appears to move or fluctuate around its average position in the crystal. And it's derived directly from the diffraction data in X-ray crystallography. That measures how the X-rays scatter off electrons in the crystal lattice. The intensity of each reflection depends not only on the atomic positions, but also on how much those atoms are moving. That's thermal motion or static disorder. And so an NMR structure is determined as a solution structure. Even in the protein data bank, you probably see it's a solution NMR structure of whatever protein you're studying. So there's no crystal involved. And NMR spectroscopy actually measures interatomic distances, or NOEs. They also measure dihedral angles and other constraints for molecules in solution. So you're not even in a crystalline lattice, and therefore there's no diffraction data in NMR to even extract a B factor from. So the positional uncertainty in the NMR structure is not derived from the scattering attenuation, but from how well each atom's position is defined by experimental restraints, such as we can look at NOE cross peaks, J coupling factors, and chemical shifts. Furthermore, if you look at an NMR structure in the protein data bank, you'll actually see that it's not just one structure. It's actually a protein ensemble of structures, and there's actually about 10 to 30 models of that protein in the NMR structure overall, right? So you still have fluctuational data, and you can still see the movement in the protein ensemble, but you don't have the B factor, which is that number that tells you how much different parts of the protein are moving, right? So regions that are well defined with many consistent restraints, these appear similar across the ensemble, and regions that are poorly constrained or very flexible will vary widely from one model to another. And so the structural variation across the ensemble serves the same interpretive purpose as the B factors. That shows which parts of the molecule are rigid and which parts are very flexible, right? Now, cryo-EM, cryo-electron microscopy, is also based very similarly to X-ray crystallography structure. So in the protein data bank, an X-ray structure or a cryo-EM structure will have a B factor per atom, and an NMR entry will not have a B factor per atom, right? But you will have that ensemble, and then you can look at the root mean square deviation of the ensemble structure, and you can get an idea of how much things are fluctuating in an NMR structure. Okay, so let's go ahead and visualize the molecule. Let's visualize the B factor right here. So I'm going to download, I've downloaded the 1HRC structure, and I'm going to open this first of all in Notepad, where I can actually just view the text file. So this is the text of the 1HRC structure in Notepad. And of course, we can just confirm that this is cytochrome C. Here's the title. We have the journal that this has, so we can look that up. And I'm going to come down here and we're going to look at the atom lines. Now, everything with a het atom, it's a heteroatom or an atom. These are all the actual atoms and coordinates in the structure. Now, residue 1, they couldn't really crystallize it properly, so they had to use an acetyl group. So they had to replace the residue number 1 with the acetyl group. That's why I have an ACE here, and it's just a CO, carbonyl, and a methyl group. That's why it's also a het atom, which is used for generally organic compounds. The protein files are actually atoms. So residue 1 is actually, this is actually residue 2, because residue 1 is actually not here. So let's just kind of look at what each row is. The first column is either het atom or atom. That tells you we're looking at an atom of some sort. And then the second column, it just counts from 1 down to however many atoms we have in the total structure. So that's the atom number. And so atom 1 is a carbon, atom 2 is an oxygen, and so forth. And then the third column, or the fourth column, I guess, this is the residue name. So this is ACE, it's a placeholder for the acetyl group, but our protein residue starts with glycine and then aspartate and then valine and glutamate. And then the next column is A, that means chain A. If there were more than one chain, we would have chain B and C and so forth. And then we have a residue number. The acetyl group is just numbered to 0 because it's a placeholder, so we start with number 1 right here. And then the next 3 columns are the X, Y, Z coordinates in Cartesian space. So this actually, the programs use this to define our coordinates. And then this column over here is the B factor. So this is 56.8. These are all pretty high at the N terminus, as well as if we go down to the C terminus, we can kind of scroll down. They're also fairly high. So this is 76 and 66 over here. In the middle of the protein, they're much lower. So this is 20, this is 10.71. We have another 20.77 here. We got 14.94. So in the middle of the protein chain, they're fairly low. On the ends, they're fairly high. So that's how we find the B factor in the PDB. And then we can visualize in Chimera X, we can visualize our protein. And we're going to load this. We're going to open. We're going to open my 1HRC protein, and this is what cytochrome C looks like. This is human cytochrome C, right? And so we see the heme group in the middle. We see this this iron atom that is in the center of the heme group. That's complexing with the histidine residue. Here's the histidine nitrogen. We've got that nitrogen, and then we've got the methionine. We have a sulfur and a methionine that are both complexing with the iron in the heme group, right? Now the ribbon shows from the N to the C terminus. That's the linear sequence of amino acids, right? And so they're typically displayed as ribbons so that we can see the alpha helices. And if there are any beta sheets, there's none in this right here, but beta sheets would also be displayed as well. And to visualize the beta factors, I go up here to Tools, and I go to Structure Analysis, Render Select by Attribute. And so that gives me this window. And from here, I just select Attribute right here, and I go to B factor. We can actually see a histogram of the B factors. And so the more blue is going to be a low B factor, the more red is going to be a high B factor, right? And you'll see that most of them, if we just kind of look in here, these are between 5 15, maybe 12, and up to about 36 or 39, and then they get, most of them are going to be somewhere in this range here between about 12 and 40. Very few are very low, and very few are very high, but we do have a couple of high B factors. And so if I just click OK, now my ribbons are colored by crystallographic B factor. The tightly packed region in the middle has a fairly low B factor, so it's blue. It gets more towards the white as the B factor increases. This end over here, that is going to be the C terminus. That's red because it's one of the highest B factors, so that's moving around. That's fluctuating a little bit more than the rest of the protein, right? So that is how we visualize a B factor in Chimera X. Now, if we use Mo, let's look at Mo, and Mo can also visualize a B factor as well. So again, we're going to load the PDB right here. And again, we have a, oops, there we go. This gives us a different view. So here's our heme group right in the middle. I'm just going to right click and I'm going to display the atoms like this. And again, we see our histidine and we see our methionine on each side, right? And if I wanted to visualize the B factors, I would just right click and go to ribbon, and I can change the color of the ribbons to whatever we want. By default, the alpha helices are in red, and the beta sheets, if any, are in gold. There's none here. And then our random coils are in white, and our loops are in blue. But if I select B factor, I can actually visualize by B factor very similarly. And so this kind of goes from blue, the dark blue, then we have purple here as it gets more red, so from purple to magenta to red and all that, that gets to be a higher B factor in the protein. So then we can use this information to study the protein in a little bit more detail. So that is what we have in our demo on B factors and how to visualize them. And so I hope you've enjoyed this video. And go ahead and hit the like button down below. Hit the subscribe button if you want to see more similar content to this. And that's very helpful. And go ahead and hit the bell icon. That actually notifies you when we post new content. And if you have any comments, leave a comment down below in the comments section, and that's very useful. I like to read those comments and see what people are thinking about all the videos I'm making. So until then, thank you for watching, and I will see you in the next video. Thank you.